past 10 now, and we've got a number of people in the room. No one else seems to be joining us at the moment, so I think we should make a start. It would be great for me if you can just confirm that you can actually hear me. That would be good. Otherwise, you always feel like you're speaking into a void. And someone can unmute themselves and maybe just say that that you can hear.
Yeah, I can hear you, Elliot.
Great, thanks very much. Okay, so welcome to our second series in our webinars for 2021. For those of you who were here before, we had spoken about remote sensing and its uses in the water industry. And today we're going to sort of go to the opposite extreme. We're going to look at the microscopic work that we do and its macroscopic uses in projects. So, Nicola, can you maybe just move on to the next slide for me? Great. So your host this morning is myself. So I'm Elliot Taylor. I'm the Divisional Director for Apeb's office in Ireland. We opened an office here in September last year, so been going about 8 months now. And one of the services that Apeb offers is biolabs. So we thought we'd talk a bit about the kind of work that can be done in the biolabs and how it's useful in a project setting. So the presentation will be made this morning by my colleague Nicola Penisi, who's our marine laboratory manager, but he's going to cover freshwater and marine. We move to the next slide, thanks, Nick. So just a brief introduction to Apeb. We're a global environmental consulting firm providing independent advice and guidance to support government and environmental guidelines. We have a range of services, as I've referred to: water, renewables, marine and ports, our biolabs that we'll talk about this morning, construction, transport, power and utilities. Nick, can you move on? Yeah, so we cover from source to sea, from the upland water areas right down into the marine environment. In Ireland, we're working a lot in offshore wind, for example, at that end of the spectrum. The next slide, please, Nick. Yeah, so we're mainly a UK-based company, although we have offices and we're working in the US, in Australia, Vietnam. Most recently, we've opened the office here in Ireland, as I said, in September last year, and we're growing that office here. We have already 2 people working in the marine field, and we hope to be adding to that in the freshwater and terrestrial fields in the short future. So I think that's it for the introduction. So what I'll do now is I'll hand over to my colleague Nick, who will talk to you about the microscopic and its use for macroscopic projects. So over to you, Nick.
Good morning, everyone. Thank you for introduction, Elliot. Thank you all for joining us today on this webinar. We're going to explore and see what is the lab work we perform at Apeb. I'd like to start with a brief introduction on the structures of our biolabs. We basically have 3 branches or sections of our biolabs that have a particular expertise, a specific expertise each. We have the Phytoplankton Laboratory, the Freshwater Invertebrates Laboratory, and the Marine Laboratory. Although these may seem distinct entities and we perform slightly different type of analysis, at the end we all work in conjunction, we all work together, and the areas of expertise and the type of analysis often overlap. Think about the phytoplankton, for example, that can be found on any kind of water bodies, from river, lakes, and marine, of course. And the marine invertebrates, the freshwater invertebrates, sorry, have a kind of a gray zone, which is the brackish and estuarine waters, or the lagoons, or some salt marsh. So again, our expertise is shared We do any type of analysis in all the labs, although we have our own expertise. Geographically, our lab are distributed all over the UK. We have, for the labs, 3 main locations. The Phytoplankton Laboratory is entirely based in Stockport, with my colleague Richard Bassett as a lab manager, although some of the phytoplankton analysis is performed in the Marine Laboratory in Letchworth, which is where I work daily. And then the Freshwater Invertebrates Laboratories, we have 2 labs at the moment. One is based in Stockport, again with Richard Bassett as a lab manager. The other one in Edinburgh with my colleague Sally Donaldson to oversee the lab. We are probably the largest, all combined, the largest lab in Europe to perform this type of analysis. And our experience, if we sum all the years of experience of each of us, adds up to more than 160 years altogether. But although we are based in the United Kingdom, our expertise is quite global. As you can see from this map, every yellow star is an area, a sea or land area that we have received samples from. Sometimes a single sample, sometimes a few specimens for identification, verification, sometimes quite a large project like a recent Mid-Atlantic deep water project that consists of more than 230 samples from about 15 to 2,000 meters deep. It's going to take too long to mention all the locations. There's a few that are fond in my memories, like samples from around Santa Helena Islands, or the Falkland Islands, the South Sandwich Islands, also the Faroe-Shetland sponge belt, which may not sound that exotic, but believe me, they're very interesting. They were very interesting samples. And then, of course, the Mediterranean samples, but I'm a little biased on that, coming from the Med myself. Okay. So if we can move on, let's see what we're gonna explore today in slightly more detail. So I'd like to introduce to you today what we do in the biolabs at Apeb, which is mostly summarizable as identification or any kind of biota to family or species level, depends on the project requirements and on the specific means a goal of the analysis itself. But also we're going to see why we do this analysis. So how and for what scopes we produce these data, which can be for support or the monitoring of a water body or of a baseline assessment, etc. And we also see how we conduct this analysis, at least on a very superficial level. We're going to see how our phytoplankton lab work on the UCAS accredited method for 17025 international standard. We're going to see how my colleagues in the freshwater labs who all held a Natural History Museum freshwater macroinvertebrate species identification qualification works on the samples, and how our marine lab operate, which our marine lab is also an appointed administrator for the North Atlantic Marine Biological Analysis Quality Control Scheme, for which we held the administration for the marine invertebrates, fish components, and particle size analysis component. The what, why, and how, they're probably not going to be in this order all over the presentation. I'm sorry. Okay, so let's start with phytoplankton. Phytoplankton is the base of the aquatic food web. I like to refer to it as the center of the food web in the aquatic environment. They are They produce most of the oxygen we breathe, and also produce that primary producer that produces most of the nutrient that then other species will eat up to the food chain, finishing with us. There are different groups of phytoplankton. The main groups are the dinoflagellates, which possess a little bit of a mobility, the cyanobacteria, also known as brooding algae, and the diatoms, or Bacillariophyceae. And most of them are buoyant, of course, so they float on the top layers of the water's surface. Some of them, like the diatoms, live more in close contact to the water body bottoms, to the sediment. We refer to this kind of organism as benthic or bentonic. They require inorganic nutrients in order to grow their cells and to reproduce, and they convert, of course, the sunlight and CO2 in oxygen as part of the process. Any changes in the environment has a really high impact on the phytoplankton, because they're extremely sensitive of changes in nutrients, pH, and other physical-chemical characteristics. Okay, in this slide, you can see on the background a, I'll say, typical phytoplankton sample and contrast phase. This is probably from a ballast water sample. But then in the two hexagons, you see two examples of the main groups of and the main analysis we perform on phytoplankton. So on the right hand side is a species of pedestrium, which is a green freshwater phytoplankton species genus, and each of these is a single cell, and they form this nice to see colonies with particular shapes and process numbers. On the other side of the slide, here we see an Diplones litoralis, also known as Navicola litoralis. There's still a little indecision on the full classification of this, but this is a diatom, and I think we can start looking at diatoms in more details now. So what are diatoms? Diatoms are unicellular algae, and they are the peculiarity of being enclosed in a silica-based cell wall, the frustule, which is much harder than any other algae or plant cell wall. They belong to the class of Bacillariophyceae and have a size range that goes from 2 to 500 microns. 500 micron is 0.5 of a millimeter. I need to keep reminding this to myself. They're extremely important in the environment. They are one of the best source of oxygen production. It is estimated that the diatoms themselves produce up to 50% of the entire global oxygen. So half of the air we breathe daily comes from diatoms. They also process silicon to incorporate in the silica base shell, and is estimated they can process up to 6.7 billion metric tons of silicon per year. And then the frustule, the dead silica shell of the diatoms, can be found up to half a mile down on the ocean floor. As most organisms, they can reproduce, most unicellular organisms, they can reproduce by binary fission, but this has a downside, or should I say a downsides. In fact, in each reproduction stage, the average size of the population decrease. Why this? The frustule is not a symmetrical structure, is formed by two halves that we call valves, and they are one of the valves is slightly smaller than the other. So when the mother cell divide, each daughter cell inherit one of the two valves, and they always they need to create the second half, and they always create the smaller half. So each reproduction stage will generate a normal size, let's say normal size cell, and a smaller producing size. And this can go on up to the point that they reach the smaller limit in the size range for the species. At that point, another type of reproduction kicks in. They form a structure called oxospore, which is surrounded by perizonia. Perizonia is still silica-based, but it's not hard test. They're more in the shape of bands, so the cell can keep growing and increasing in size up to when they reach the maximum size range for the species. And at that point, the binary fission reproduction will start again. Okay. So in our phytoplankton labs, we process an excess of 2,000 samples per year on average, including the really weird 2020. From all around the globes. When I mentioned the 230 samples from the mid-Atlantic, there was the benthic side, but we also had about 150 phytoplankton samples from there. In order to process the diatoms to improve identification, we digest the sample in a hot bath of hydrogen peroxide, about 90 degrees Celsius, in order to remove everything that we don't need for identification. So we remove chloroplast, cytoplasm, everything else but the first two. Then after the digestion, we use a centrifuge to separate the digested material from frustool, and we proceed with identification of all the frustules on permanent slides, identification and enumeration, of course. At that point, the frustule has probably been divided in valves, so we're going to identify the single valves. What kind of information we can understand from diatom samples? Well, the diatoms, as all the phytoplankton, are very sensitive to changes in environment, as already said. In particular, they are very sensitive to increase of variation of inorganic nutrient concentration in the water. We use the trophic diatom index to assess the level of nutrients in the water. Basically, each species have an assigned score against this sensitivity to the change in the concentration of nutrients, and then we weight this score all over the sample based on the abundance of that particular species or group. This way we can calculate the trophic diatom index. The higher is the index, the higher is the inorganic nutrients concentration in the water. And this can be for any kind of reason. It can be for any kind of discharge, pollution incident, runoff from an agricultural land, wastewater discharge, any type of event that will input into water more nutrients that are naturally occurs. Diatoms are also very sensitive to change in the pH, so they are good gauge to check the acidification of waters. And in this case, we apply the similar method, but using the acidification sensitivity to score them, and we calculate another index called diatomic acidification metric. Sorry. So we can also we're also able to, by collecting paleocores of sediment, of gather information about historic condition, because the Diatom frustulase is very good on fossilization and is very resistant, and therefore we can gather information on historic condition up to hundreds and thousands of years ago. The other side of the phytoplankton analysis is to analyze the entire sample. So if you remember the slide I showed you earlier, the slide, the yellow one, that entire sample was both diatoms and other type of phytoplankton. In order to do so, we cannot proceed with the digestion, otherwise we know any other cell but the diatoms will be dissolved. So we need to use a slightly different approach. The first thing we do is the homogenization of the sample. It's a fancy word to say that we try to distribute any phytoplankton cell across the sample evenly as much as possible. And this is performed by a particular set of movement on the sample bottle itself. Once has been homogenized, the sample been transferred to sediment chambers, which are this chamber you see in on the slide, and they are allowed to rest in order for the phytoplankton to settle down on the bottom of the chamber. So the sample are then transferred to an inverted microscope where we identified and enumerate the organisms. Sometimes, especially for freshwater sample, we also perform measurement of the cells in order to calculate the biovolume of the plankton. So why we perform this kind of analysis? Well, of course, the phytoplankton studies are indispensable to the water frame directive and calculation of ecological quality ratio. But we can also use this type of analysis to identify any kind of toxic nuisance in the marine environment, but also to manage the algal blooms that can occur in water reservoir or lakes. We also are very active on the ballast water front. We regularly analyze ballast water samples in order to spot any non-native or invasive species that can come to the UK or to the area from where the sample come from, and so stop the spread or at least monitoring the condition of the new introduced species. It is also a service quite request for lake and reservoir management because of the algal blooms most of the time, and opens water and swimming leisure activities as well for the same reason, and basically everything under the Bathing Water Directive of 2006. The algal bloom is a particular important topic because it becomes an increasing problem, at least across the UK. Basically any change in condition, nutrients, acidification, sunlight, exposure, temperature, can spark an excess growth in the population of the phytoplankton, in particular of the cyanobacteria. Now, cyanobacteria produce a toxin called cyanotoxin that can be really dangerous to the wildlife, to other invertebrates, to fish, or even to small mammals, also to the human beings in the worst case scenario. So it's something that needs to be monitored constantly and managed. Symptoms of the cyanotoxin poison can be various, from a simple skin rash to a severe liver disease. Okay, these are some examples of a really few species or genus of phytoplankton we can find in our sample. So on the top left, we have a representative of the genus Anabaena. Anabaena are estuarine brackish water species. They can produce toxins, and they form this kind of a pear necklace look like colonies. On the bottom left, we have a representative of a colonial diatom from the genus Chaetoceros, with this very, very nice shape of the colony and production from the first two. We have a representative of a green algae on the bottom right corner, Cosmarium sp, and then probably my favorite on the top right corner is what is called sea spark, scientific name Noctiluca scintillans, which is a bioluminescent phytoplankton responsible for some of the beautiful, fascinating photo you've seen all over the news and social media of the blue sparkling water in the night all over the globe. The Freshwater Laboratory is another strong branch of our company. We process about 1,600 samples per year on average, again, including 2020, which was an anomalous year for reasons that we all know. Our method is based on the Environment Agency standard method, the freshwater microinvertebrate analysis or riverine sample from 2014. This means that all our samples are sieved over a 0.5 millimeter mesh. Samples are collected from rivers and lakes, sometimes using small graphs, more often using the standard approach of the 3-minute kicks and hand search for various habitats, all the possible habitat present. Transferred to the lab, they have a very fast turnaround for analysis as the method imposes. And they're most identified to family level because of the water framework directive metrics that we need to calculate. We can take them to species. We often do this regardless of the requirement, especially if we need to look for invasive or non-native. And so they're used to calculate the ecological equality ratio that has 5 classes, from high, which is an understood ideal environment, to good, moderate, poor, and finally bad condition, the condition we don't want to reach ever. There's more or less for phyla 7 classes and hundreds of species of freshwater invertebrates in the samples we routinely analyze. And each taxon has different ecological requirements and tolerance to changes in the environmental condition. So we tend to divide them in 3 main group. They are tolerant, intermediate, and sensitive. Sensitive are the species that don't like at all changes in condition. And the EQR is mostly based on this diagram, which is an historical diagram from Heinz 1960, the biology of polluted waters. May look complicated, but it's quite simple to interpret. Basically, these 2 halves show the same river from a physical chemical point of view and a biological point of view. This outfall is the pollution event we will take in consideration. Everything on the left-hand side are ideal conditions. So we see from a physical chemical point of view, before the outfall, we have high level of oxygen and very low levels of suspended sediment, salt, and biological oxygen demand. On a biological point of view, we have high level of extremely sensitive species that want and live in clean waters, and very low level of any other species, either intermediate or tolerant. Once an outfall event happens, we see a drop in the oxygen level and a sudden spark in the biological oxygen demand and suspended solids and other nutrient distribution. On the biological side, contemporary, we see there's a drastic drop in the sensitive species abundance distribution up to the point that they can disappear, and an increase on the tolerant or intermediate species. A little bit like the Diatom trophic index, we assign to each relevant taxon a score based on their sensitivity or tolerance, and we also consider the number of scoring taxa all across the sample, and by interaction of these two numbers, we calculate an index that then is translated in the high, good, moderate levels that we've seen earlier. We also keep an eye always on the invasive and non-native species, because it's imperative that they be monitored closely to avoid any uncontrollable spread. Some example of all these groups, the stonefly larvae are a good example of pollution-sensitive organisms, particularly Brachyptera putata is an example, but there's much more. While the pollution tolerance can be the larvae of the Siphidae or flower flies, overflies, and their particular shape for which they call rat-tail maggots. What to mention among the invasive species, the quagga mussel can be a big nuisance for a lot of reasons, including the man-made structure that can be clogged by them, and also what made the news a few years ago, the demon and killer shrimp. These two organisms are both belonging to the Dicarogamorous genus, and they are extremely voracious predators. Actually, sometimes they don't kill even to feed themselves, just to take the territory or maintain their territory, and they can kill everything in the water body, from other invertebrates to small fish. So they can be extremely harmful for the native species. And to distinguish between them, it's quite hard, and as usual, the devil is in the details. So the difference between the two species at the end, it's just the number of spine on the urosome segments, whether it is 2, 3, or more. And now we can come to the marine laboratory. As I already said, we are probably the largest marine laboratory in Europe performing benthic analysis. Our samples and our analysis include not just the benthos, though, and not just invertebrates. We work on meiofauna samples, zooplankton. We process marine phytoplankton samples. We identify fish from the egg stage to the larva stage to the adults. And we also do analysis of macroalgae. When a sample comes to the marine lab, everything in the sample will be analyzed. We are the contractors for the NMBA QC scheme for the invertebrates, the fish component, but also for the particle size analysis. We work for all the major agencies, environment agencies, both in England, Scotland, Wales, Northern Ireland. So most of the regulatory authorities, conservation agencies, but as well for construction companies, water companies, power companies. Big part of our job is related to wind farm energy or offshore oil and gas extraction, but also aggregates, and we also do analysis for other consultancy companies. Collecting samples from the marine environment is slightly more complicated than the freshwater, mostly because some of the depth we need to reach. So we do quite a lot of intertidal sample, both for phase 1 and 2. Phase 2 involves collection of sample for further investigation, and this is made by the insertion of a core sampler in the sediment in order to collect a specific volume of this sediment to be analyzed in the lab. Everything subtitle needs to be taken using drop-down video on top video, for which we perform analysis as well. But most of our sample, of course, are physical, and they're taken by devices called grabs. These are three examples of the main example of grabs we use in our service. This is a day grab that is mostly used to collect samples from muddy, silty environments up to a certain depth, of course. Then after that depth, we need to start using subtidal course systems. For example, the day grab is used often for samples from the Thames, which is a very particular environment. When the particle size increase a little bit, so we pass to fine to coarse sand, it's better to use a Van Veen grab. And finally, when the sediment size is mixed or large, like gravel, cobbles, or any other kind of large type of sediment, the Hammond grab is the best device, although we use a mini Hammond grab in order to have a comparable size to the day grab here. Once the samples are collected, they've been sieved on board to the required mesh size that is either 0.5 of a millimeter or 500 microns or 1 millimeter, fixed in formalin before being preserved in IMS back in the lab, and transferred to the lab for analysis. In the laboratory, then we proceed with extraction of all the biota, both fauna and flora, and identification on the microscope. We analyze an excess of 2,500 samples per year on average, and this is just to include macrobenthic and zooplankton, so I'm not considering this count any phytoplankton or analysis or video analysis. The proportion is about 1,500 macrobenthic and 500 each zooplankton and ballast water samples. But for ballast water sample, we do both phytoplankton and zooplankton analysis, mostly for early detection of any potential invasive or non-native species. Our method is mostly based on the NMBQC guidelines that incidentally have been written by two Apeb senior staff more than 20 years ago, and are up to date, the standard for the British industry and, well, I'll say Northern Europe industry. But also some of our procedures, especially on the quality control, are reflecting the International Standardization Organization method, ISO 16665, latest update of 2014, which is called Guidelines for Quantitative Sampling and Sample Processing of Marine Soft Bottom Macrofauna. Some of the details that we need to identify our biota require move the sample to high-power microscopes with higher magnification than the compared to the dissection microscopic seen here. This is because, contrary to the freshwater and the Diatom phytoplankton analysis, they are not, we don't use indexes we use statistical analysis mostly based on biodiversity. So we need to know as best as we can how many different taxa are present in our samples and their relative abundance. So we need to take the identification to the lowest taxonomic level possible, and therefore we need high magnification microscope. We will see later some details of this kind of analysis. As the good laboratory practice dictate, we held a reference collection. Our reference collection wall is quite extensive. We currently have an excess of 7,400 single entries in our reference collection, and this can be used for any kind of reason. It's just a historical record of the project we have worked on, but also it is very helpful in identification of difficult taxa. And often we we lend our specimen, genus or families, depends the request, to international experts that are working on expanding the knowledge on that particular group. We also held an extensive literature, especially related to the foreign samples that the overseas samples that we analyze. Oh, almost forgot. I'd like to point out also this structure on top of our dissection microscope. This is a microscope camera that was used to take in this photo and all the other photos you see through this presentation. All the photos in this presentation are Apeb, made by us by a technique which is called stacking. We need to take photo at different magnification and a different focus, and then with a particular use of software and human skills, we stack them all together to form almost a tridimensional photos. And these photos can part of our service. We can offer photographic reference collection to our client. It can be used for just cosmetic of the reports, as well as marketing purpose, as you've seen if you have a look on our social media. Or as we do in the lab, it's just nice to see hanged around the walls. So, what we can see in a marine sample. This is a typical microscope where we have our petri dish from which we extract the smallest part of the biota. The largest part is extracted in trays, especially because in the marine environment, the organisms tend to grow on each other, especially the epibenthic. So it needs a certain level of experience to separate them in trays, then transfer them on a microscope for further sorting. The four major taxonomic groups that can be found in marine environments are shown in this slide. There are polychaetes, crustaceans, mollusks, and echinoderms. There's a fifth group that includes everything else. In fact, it's called others or miscellanea, and includes any other kind of non-bristle worms, such nematodes, nematodes, flatworms, but also any other kind of arthropod which is not a crustacean. For example, insects or arachnidae or sea spiders, which are their own group. And by the analysis of the combination and the biodiversity of this species, we are able to understand the biotope of that specific area and build up biotope maps of the seafloor or the shore. One of the main groups, polychaetes and crustaceans are the main groups we found. Polychaetes probably have more than 8,000 species all over the world, while crustaceans have even more, about 65,000, more or less. Mollusks are also widespread with about 85,000 species all over the world, and finally the echinoderms. These isopods come from the ferrospongial seabed. It's quite, let's see if we can expand it a little bit. There we are. So this is Toella laciniata, an isopod. Isopods are living mostly in aquatic environment, but we can find some example of isopod terrestrial, the understones, earthpigs, I think it's called, that you see in your garden, and they are the only group of terrestrial crustaceans, the isopod. Polychaetes are cosmopolitan, you can find them everywhere. This Neremia punctata belongs to the family of Esionid, and it was the first Esionid ever identified back in 1776 by Müller. This bivalve, Theora lubrica, is a non-native species that we recently found in Lowestoft in England. This is a photo from our paper from the description. Sorry, when we found the first specimen in the UK. It's native of the West Pacific around the Sea of Japan, and I moved then from Japan to New Zealand to the US, arrived on the European coast of Netherlands some time ago, and it is now recorded in Britain as well. And finally, this is a representative of a sea cucumber from a deep sample in the mid-Atlantic. It's probably an undescribed species belonging to the genus Smolpedia that I'm currently investigating. These are some more examples of polychaetes that, as I said, are ubiquitous and have a lot of different families, species, and of course shapes and habitat and behaviors. Again, these are just few examples of worms that I like a little more than others, but for no particular reason. Spirobranchus is a representative of Serpulid family. The Serpulids have this characteristic of being bicostructure. They build calcareous tubes. They can form a structure called trottoirs that you can see in the Mediterranean. So it's a large bicostructions. Largest coreni is a representative of the pectinarid groups, the family, sorry, also known as ice cone tube worms or trumpet tube worms because of the shape of the tubes they construct and the characteristic of this golden chaetae or around the oral area called palii. This one is a representative of the polynoids, also known as scale worms, and they have modified some of the bristle or chaetae in scales that they use for camouflage and protection purpose. They are mostly active predators, although they tend to stay hidden to avoid to be predate themselves, and they can be found living in symbiosis with other tube-dwelling or burrowing organisms. And finally, this rather ugly worm is a representative of Sternapsidae, which is recently been reviewed as a family, and this genus was considered to be cosmopolitan, but of course, as every cosmopolitan species, they're probably not. In fact, much more species have been described recently, and they are very difficult to identify. The details to be looked at the caudal shields here. They live burrowed in soft sediment, and they use this shield to close the burrow, and when they need to breathe and feed, they exit from the burrow with the gills and the tentacles from this area, which is not the head. This is actually the tail or anal end. The head of the worm is here, and it can be entirely retracted inside the body. This structure is called introvert. It can be entirely retracted, so they become even more protected from predation and disturbance. Some example of crustaceans with representative of the main groups we can find in our sample. This is Acomesha, Campylapis orida. They also call comma shrimps because of their shape with this enlarged body and kind of thin tail. This is a representative of a tiny insides group, Tanaids, Pseudos laterally. They can be found in any kind of environment from intertidal to very deep sea. Amphipods with Dexamine spinosa as an example are probably the most abundant crustacean we found in benthic samples. We will see shortly where we can find it in plankton samples. And finally, a representative or slightly more evolved crustacean belonging to the known group known as Mantis Shrimp. This is representative of species you can find in Britain, Rissoides desmaresti, but in the Mediterranean, the most diffuse one is Squilla mantis, which is also commercially important as edible species. In the zooplankton samples, the most abundant group at all is the copepods, which again belongs to the crustacean. They're very small, and they're ubiquitous, and someone consider them to be the largest animal biomass on Earth. Some might argue they are euphasids, so there's a little bit of a debate, and as you can imagine, it's not easy to solve this dispute. I expect more study to come on this topic very soon. This is just a generic calanoid copepod. It's very hard to identify just by these details. In order to identify, you need to check some of the legs and the caudal structure, and require quite a lot of magnification. Here's some mollusks, the four main groups of mollusks. From the more primitive, the chitons here, where the shell is divided in the separate parts or valves, and they live in close contact to other substrata, so they can, a little bit like limpets, they can just adhere to the surface and be protected from predation. But then when they start moving around this, the animals lift from the bottom, and this girdle will appear. This is a soft part of the animal to move around, and underneath we have the gills and the mouth structures. These are two representative of gastropod, two different groups of gastropod, opistobranchs and prosobranchs. The difference stays in the position of the internal structure and the characteristic of the mouthpieces called radula. And finally, representative of the bivalves with Thyrisara flexuosa, which is widespread all over the North Sea. And here we have some echinoderms, probably my favorite groups. My favorite group, the starfish. We all know starfish. This is a tiny species, Asterina gibbosa. This is 2 to 3 millimeters. It doesn't get much bigger than this. Strictly related to starfish are the brittle star. This is Anophira alba, widespread all around the British Isles. They are a lot in common with starfish. This is a oral view, so this is underneath the animal where the starfish, you can see it from the dorsal view. So the mouth is underneath them. They move with the mouth in contact with the seafloor, so they can feed themselves, and have the characteristic of this very elongate and serpentine shape arms, very fragile, hence the name brittle star. This is a representative of sea urchin. In particular, this is an irregular sea urchin or sea potato, commonly known, where the pentameric symmetry has leave place to a more bilateral symmetry, although the pentameric symmetry is still present in internal organs and in the petals here on top of the dorsal side. And finally, a sea cucumber, where the pentameric symmetry or bilateral symmetry is mostly gone, but it can still be seen by the distribution of the structure around the body, and more important, internally. I mentioned the other group of miscellany, where all the minor groups are included. They're not minor because they're smaller, they're minor because they are less present as an average in our sample, but still very much present. Here's some example. This is a sipuncula, although it's wrong to be included in this slide because there has been recently moved. The anti-sipuncula phylum has been moved to the polychaetes, as some internal chaetae has been found recently, well, more or less recently. They are commercially important in some part of the world. They are considered prelibacy in Asian food, not this species, but others, of course. This is a representative of what is commonly known as moss animals, bryozoans. This is an erect branching species, but most of them form like a carpet covering stones and hard surface on the in the sea bottom, and they are colonial. That's each one of these little white dots you can see is a single zooid. This is a colony of sponges belonging to the genus Leucosolina that form this quite intricate lattice. And finally, the infamous Nematostella vectensis, which is considered both invasive and protected species because was originally found in a lagoon environment, but is not limited to lagoon environment. So there's this duplicity role of being invasive, but also being protected by law. So what kind of really microscopic detail we're gonna look to identify our biota? Do you remember this? I showed you this a few slides ago, and it's a species I'm currently working on, is a Molpadia from the deep ocean floor in the mid-Atlantic. In order to properly identify this animal, we need to see the internal structure as well as some details of the skin. These kind of geometrical shape structure you see are called spiculae, which give the name to the class of decanoderms. That means spiky skin. And by the shape, distribution, and difference in the spiculae, we are able to restrict and identify most of the sea cucumbers. Peculiarity of the Molpadid family is that some of the spicule modify and becomes pigment all across the skin, these red patches. When we identify polychaetes, most of the times the details we need to look are the chaetae, the bristle that emerge from the parapods, lateral parapods in the worms, and we need to go really in deep details. Now the chaetae can have a lot of different shapes and functions. These are just a couple of examples. This is a circular chaetae that held together the parapod and its auxiliary to the movements. With these are the called Falchiger and Pseudospiniger chaetae, for which we need to check sometimes the number of teeth or the distribution and shape of the teeth. So we need to go quite in deep details. Consider that this is the scale is 0.1 of a millimeter, and this even high magnification. For the crustacean, although most of the time we can identify them using the general body shape and characteristics like spine and some details on the legs. More often, especially for some kind of shrimp, we need to go in very deep details. This is a photo from a scanning electron microscope of the mandible of a Macrobrachium nipponensis, Asian Japanese shrimp that can be identified just using the structure of the mandibles. If then go in further details, we'll see that each of the scospid is not a single structure, but is formed by a lot of bristles close together. It's a really fascinating word, and I hope you enjoy the presentation. Thank you.
Thanks so much, Nicola. That was absolutely fascinating. I'm from a freshwater background, so your journey into the marine environment was really very interesting. I think maybe you've got the more interesting beasts there, but I've always been a fan of the freshwater.
I'm afraid I'm a little biased on the marine side. I'm sorry.
So we've done an hour. So some still people who are still with us, which is great to see. I don't know if anyone has any questions. You should be able to unmute yourself if anyone has any questions that they'd like to ask at this point. Okay. Well, I think maybe you've been very comprehensive in your explanation, Nick. So that's great. So I'll just finish by drawing your attention, please, to our next webinar, which will be on the 2nd of June, where we will be talking about hydromorphology in Ireland, issues and solutions. So again, something very relevant to achievement and implementation of the River Basin Management Plan and achievement of Water Framework Directive requirements. So I look forward to seeing you or some of your colleagues then. We will send out notification of this nearer to the time. And if you have any other questions that you didn't get to ask now, or you have any interest in the kind of work that we can do on microscopic work for macroscopic projects, then please let us know. So with that, I'll say thank you very much. Enjoy your day. Thanks again to Nick for your fantastic presentation.
Thank you very much for everyone to attending, and to you, Elliot. Any question, please let me know. Thank you. Goodbye.
Thank you. Bye-bye.